Hi, Miss Nikki here. Welcome to Chapter 2, Part 2. We're going to talk about some of the biomolecules that make up organisms. So we're going to talk about carbohydrates. We're going to talk about lipids or fats. We're going to talk about proteins. And we're going to talk about nucleic acids. So we have to talk about these biological, macro, or large organic molecules. They always contain carbon, hydrogen, and oxygen. So we're going to talk about the four classes that I just talked about: carbohydrates, lipids, proteins, nucleic acids. They're always going to contain these three elements, and then they can also have these three elements. So if you remember the chinops that I talked about earlier. And remember that carbon has 4 valence electrons, so it wants to bind with 4 other atoms. And that allows for these carbon skeletons to have a variety of forms and be really versatile, right? We saw some that formed rings, some that formed these really long tails of carbon atoms bound together. So let's go through and talk a little bit more about each one. Before we move to the individual topics, we first have to understand how we link them together. So we have this word polymer many and monomer one. And what I would know is which one's the polymer. These are all polymers. And then which ones are the monomers and which makes up the polymer. So if you have sugar molecules, so if I have sugar molecule 1 and sugar molecule 2, and I put those two together, I'm going to have a carbohydrate. For nucleic acids, the monomers are nucleotides. So if I have nucleotide 1 plus nucleotide 2 and I join them together, I'm going to have a nucleic acid. And then the same with amino acids. If I have amino acid 1 and amino acid 2 and I join them together, I have a protein. So the process by which this happens is called dehydration synthesis and hydrolysis. So these are reactions that are going to occur to either join monomers or to pull apart polymers. So dehydration synthesis, most of us know this term, right? Dehydrate, remove water. So here in dehydration synthesis, I have monomer 1 and I have monomer 2, and I want to join them together into a polymer. Do you see how they removed the water, and that allowed for the bond to form this dimer, and now you have a polymer. So dehydration synthesis is what happens if you want to take one monomer and another monomer, and you want to join them together to make a polymer. So hydrolysis, hydro, water, lysis, split. So here's hydrolysis. We have this same dimer. We want to split it back into its individual monomers. So we insert water. We're basically doing the opposite of what we did in dehydration synthesis, right? So we push water in here. It separates the dimer, and now you have monomer 1 and monomer 2. So hydrolysis is if you have a polymer and you break it down into one monomer plus another monomer. And why would this be important? These two processes are very important because this is how we can store food for use later. So the example I usually give is if you come home late at night, you've been partying all night, you're starving, you stop at the drive-through, whatever's open, Taco Bell, right? And you get something to eat and you scarf it down in the car, you get home, you climb into bed, and you go to sleep. So what happened to all the food that you consumed, right? You're going to store it, right? So you're going to take those sugar molecules, those carbs, and you're going to store them as a polymer, and you're going to store them for use later. You wake up the next morning, you feel terrible, you think the best thing to do would be go for a run. I'm going to go for a 3-mile, 5-mile run. So you throw your clothes on, you head out the door, and you run and you run and you run. You didn't eat anything. Where did you get the energy to run? Well, all the stored food that you ate the night before, we do hydrolysis, we break it into individual monomers, and then those monomers can be used to fuel the body. And we'll talk more about ATP as we move through this PowerPoint or the next. So without dehydration, synthesis, and hydrolysis, you cannot store food for energy later, and you would have to eat every time you wanted to do work. So that wouldn't be very good, right? First macromolecule or lipids, basically we should be thinking fats. We're going to talk about these 4 primary classes, you should know each one of these and just have a general idea, like one statement about what function they serve in the body. So triglycerides is that first classification we'll talk about. You might have heard saturated, unsaturated fats. So this is your long-term energy storage. So this is adipose tissue, right? Fat tissue. We'll talk a little bit more about this technical term adipose when we get to histology. And again, you can store triglycerides or break them down. So lipogenesis, this is creation of triglycerides. Lipolysis, the breakdown of triglycerides. So you do need some triglycerides so you can have this long-term energy storage. You just don't want too many. Phospholipids are the second class of lipids. These are really important because these create cell membranes or plasma membranes. So if you take a phospholipid, it has two tails. If you take a bunch of phospholipids and put them together, the heads are going to face each other and the tails are going to face out, and it forms a barrier that separates the outside of a cell from the inside of a cell. Now we're on to steroids. This is also a lipid. We're on our third type of lipid. It'll become a little bit important when we get to reproduction chapter, so you'll come back and talk about steroid hormones like testosterone and estrogen. For us, we probably just want to focus on the cholesterol. So cholesterol is a lipid that's wedged in between our plasma membrane. Remember, this is phospholipids. So really you have two kinds of lipids making up your cell membrane or plasma membrane. Fourth group is this eicosanoids. We're really not going to come back and talk about these till 202. When we talk about inflammatory response, we'll come back and talk about prostaglandins. So some of these are going to come into play a little bit later. So for right now, as long as you say they have something to do with inflammatory and immune response, I'm good with that. So I think this is probably the number one important macromolecule. Again, that's just my opinion, right? If you don't have lipids, you can't have a plasma membrane, so that's pretty important too. But this is kind of the fuel for our body. Without carbohydrates, we'd have to work really hard to extract energy from fats and proteins. So carbohydrates, this is kind of the chemical formula, and where N is the number of carbon atoms. So let's say glucose, you'll see me write this. It's the same as if I put it in this form, like this. So glucose, fructose, they all have 6 carbons, 12 hydrogens, 6 oxygens. There's monosaccharides, that's a simple sugar. You have disaccharides, this is when you have 2 monosaccharides coming together. So if I took glucose plus fructose, these are both monosaccharides, so one monosaccharide plus another monosaccharide. If I put them together, I have a disaccharide, and we call this sucrose. Are you seeing a pattern with the ending, right? All sugars have an ose at the end. And then a polysaccharide is when we take many, many, many monosaccharides and join them together. These are 2 terms you should be very familiar with. They're going to be used throughout 201, throughout anatomy, throughout any allied health field. So glucose is a simple sugar, a monosaccharide, and it's the sugar that we use for the majority of our energy. So we're going to talk about cellular respiration, and we're going to talk about how we break down glucose and we pull energy out of it. And we use that energy to do work in the body. Glycogen is how we store glucose. So remember when we talked about dehydration, synthesis, and hydrolysis? Well, if you eat a whole bunch of food at night, you're going to take that glucose and you're going to store it as glycogen. And that glycogen can be stored in the liver and in your skeletal muscles. Any excess glucose, if it can't be stored in the muscles of the liver, then it can be stored as fat. I think this is a nice representation. So you get to see glucose, the monosaccharide, and then you can see over here glycogen. which it's a whole bunch of glucoses linked together, right? And they form these kind of branches. So if I want to break down glycogen, it's called glycogenolysis. So if I want to just cut right here and I want to have just one sugar, that's glycogenolysis. Glycogenesis is when I take a whole bunch of glucoses and link them together and create glycogen. So remember what I said? Dehydration, synthesis. Dehydration synthesis, you're taking monomers and monomers, there we go, and you're turning them into polymers. And what's hydrolysis? When you have a polymer and you're breaking it down into individual monomers. So they just changed the name so that you know exactly what hydrolysis process we're talking about. So glycogenolysis is taking glycogen and breaking it down into individual glucoses, but it's the same process. It's this hydrolysis, this split with water that we saw earlier. So I wanted you to see some of the structures of these other simple carbohydrates. So this one here is glucose. They don't put it up here. I don't know why, but they don't. So galactose, fructose, glucose. So glucose would be body sugar, like for humans, right? Animals. Fructose would be fruit sugar. Galactose is sugar you have in milk. So the reason they're showing you these two side by side is that these two are going to come back later when we talk about nucleic acids. So remember the four macromolecules: carbohydrates, lipids, proteins, and nucleic acids. So the basis of your DNA is a carbon sugar. So we are carbon life forms. Every single one of those four macromolecules, carbohydrates, lipids, proteins, nucleic acids, they all have carbon in them. So we're going to see these ribose and deoxyribose when we talk about nucleic acids. Their backbone is still sugar. They are 5 carbon sugars instead of 6, which glucose is 6. So then if you link them together, these are disaccharides. So we just took 2 monos and put them together. So glucose and fructose, and you get sucrose, which is table sugar. And then if I take a glucose and a galactose, I get lactose, which is milk sugar. And then if I take a glucose and a glucose, I get maltose, and that's kind of the sugar from grains. Now we move to our third macromolecule, and that's nucleic acids. So we just saw these sugars, deoxyribose and ribose. So the backbone of your DNA is sugar. Let's look at a nucleotide on the next slide. So every nucleotide monomer consists of these 3 components. So there's always a sugar, then there's a phosphate group, and then there's a nitrogenous base. So we'll talk about some of these bases in just a second. So the backbone of DNA is a sugar. And remember, a nucleotide, if I have nucleotide 1 plus nucleotide 2, that I put that together and that forms a nucleic acid, which is the polymer. Here are the nitrogenous bases. So we have the pyrimidines and the purines. So you can see that the purines have 2 rings where the pyrimidines just have 1 ring. And it tells you down here whether these bases are in DNA or RNA. So in DNA, you have A, T, C, and G. And then in RNA, you're going to have A, U, C, G, or GC. Some people say them in a certain order, so once you do that, you can't help it, right, for the rest of your career. So kind of the basic way to think about these are DNA is going to copy itself into RNA, and then the RNA is going to be processed into a protein. So the DNA stays inside the nucleus. The RNA is the copy that goes out into the big bad world of the cell, and it gets translated into amino acid sequence, and then eventually that gets folded and turned into a protein. Okay, so who cares? Well, proteins are going to go out into the body and do work. So we'll talk a little bit more about these next. But hopefully you can see that kind of everything builds, right? So what's the core component of DNA? What's the backbone of DNA? Sugar. What else do we use sugar for? We use sugar for energy, right? How do we get things done in the body? Proteins. Well, do we need nucleic acids? And do we need sugars in order to make proteins in order to do work in the body? Hopefully we're saying yes. So this is all interconnected. RNA usually occurs in single strands. So you can see it's got the U. What I think is interesting is you can automatically tell when looking at these two which one's DNA, which one's RNA. If it has a U, it has to be RNA. It cannot be DNA, right? If you see a T, it has to be DNA. It cannot be RNA. So think of it as this is the template, this is the copy. We don't want anything to happen to our template. We just want to churn out copies. But our template stays as original or as protected as possible. Your DNA template stays in this double helix form as well, this double strand twisted structure that you see. ATP is the third nucleic acid we're going to talk about. So remember, we already talked about DNA and RNA. ATP is also a nucleic acid. It's composed of nucleotide. It has a nitrogenous base, it has a sugar backbone, and it has 3 phosphates. If you remember, nucleotides that we saw previously only had 1. So ATP is adenosine triphosphate. This is a really important energy molecule for the body. So when we get to muscle contraction, we'll talk about the fact that we need lots of ATP in order for a muscle to contract. So basically to breathe, to think, so all neurons, all muscles, including your heart and your intestines, they all require ATP in order to work. And where do we get ATP from? We break down glucose. So we break the bonds of glucose, we strip the energy out, we store that energy in ATP, and then the ATP can be shuttled in the body, and then we can break these phosphate bonds and we can release energy. And that energy allows for work to happen inside the body. The last macromolecule we have to talk about are proteins. So I just want to do a recap real quick. DNA, right, that's your template. DNA is transcribed or copied into RNA. So RNA is the copy. And then RNA, basically this copy has instructions for building proteins. So without RNA, you can't build proteins. Without DNA, you can't build proteins. What do these proteins do for us? Lots of tasks here, lots of functions. This is probably the most important. For us, we're going to talk about enzymes. So basically, an enzyme is a protein with a specific job. And we'll talk a little bit more about these coming up in chapter 3 or 4. So you remember how I just talked about dehydration synthesis and hydrolysis? You don't just sprinkle water on something and automatically it splits a polymer, right? An enzyme, a protein acting as an enzyme, actually makes these reactions happen. So without the enzyme, those reactions won't occur. Proteins can also facilitate transport, they can offer support, they can cause movement, they can store or act as storage molecules, right? We can see in these images, if I take amino acid 1 and amino acid 2, and I want to join them together, this is going to happen through dehydration synthesis. And now I'm going to link these two amino acids together. So that is a polymer, and the polymer is called a protein. Sometimes you'll hear it called a polypeptide as well, same thing. So then we just keep linking these amino acids together until we receive a stop message, basically, and then we know we're done building the protein. If we were going into biochemistry, then we would need to know all the amino acids and the different groups that they belong to, like nonpolar, polar, charged, and some that have these specialized functions. I want you guys to understand that amino acids are linked together. The other slide, it mentioned that's called a peptide bond. So amino acids are linked together and they form proteins. And then tell me the function of proteins. What can proteins do for you? Why do you care if we have proteins or not? So depending on what category of amino acid, those polar, non-polar, you're going to see the protein kind of conform or go through a conformational change. So you can have one end interacting with a different section of the protein. So these are all beads on a string, right? These are all individual amino acids. And what happens is if the string gets really long, it can start folding on itself. And you'll have interactions that'll occur between the top string and the bottom string. But if we just have beads on a string, that's called a primary sequence or primary structure. So if you just have beads on a string, primary structure. If the string forms either this ribbon, this alpha helix, or if they line up and we have connections between the top sheet or the top string and the bottom string, we call these secondary structures. So it's not just amino acid beads on a string, but they're actually interacting with other amino acids because they're close to each other. Now we have a tertiary structure. So we might have beads on a string. That was primary. And then we might have an alpha helix. That was secondary structure. Or we might have a beta sheet. That's also secondary structure. So if I have primary structure and secondary structures all together in the same protein, we call that a tertiary structure. Now we have the quaternary structure. So sometimes you'll see it abbreviated like this, or they'll show that. So it says tertiary structure. There's also a picture of a quaternary structure in your book. Basically what you're taking is you're taking this quaternary structure and another and another and another, and you're putting multiple tertiary structures together. So you'll have interaction between this amino acid and the one that's over here. And you'll have interaction between this amino acid and this one that's over here. So basically a quaternary structure is just linking multiple tertiary structures together. There's a better image in your textbook. I just don't have the image here. So these are important. A lot of people kind of brush over the protein structure part because they're like, okay, so what, who cares? But these are really important. So hemoglobin is a quaternary structure. Collagen is a quaternary structure that we have in the body. If you didn't have hemoglobin, you couldn't carry oxygen. If you didn't have collagen, you wouldn't have tendons, you wouldn't have joint cavities, you wouldn't have, dermis, right? Skin. So it's important to understand how these are all stacked together. So if you have a quaternary structure, it's multiple tertiary structures joined together. If you have a tertiary structure, it's multiple primary and secondary structures. If you have a secondary structure, you're going to have an alpha helix or a beta sheet. And if you're a primary structure, it's just beads on a string. And this is how we decide what this protein is going to do. So what do I mean by that? What happens if you're missing one of these? Let's say your hemoglobin, and instead of having 4 tertiary structures linked together, you only have 3. What do you think is going to happen to the amount of oxygen that you can carry in your red blood cell? It's probably going to go down, right? So protein structure matters. If you change the structure of the protein, it disturbs the protein's activity. It's usually irreversible. Once you fry an egg, can you turn it back into a baby chicken? No, right? Also, pH changes. So this goes back to the first part that we talked about when we talked about blood, and we talked about it being kind of that neutral pH, right? 7.3, 7.4 area. So proteins are really sensitive to temperature changes and pH changes. So the structure of the protein matters, how it's folded matters. Whether it's exposed to increased temperatures or differing pHs, it can make this unwind, unravel. And then also the amino acid sequence matters. It depends how you link up those amino acids. So if I do 1, 2, and 3 in order, I'm going to have protein X. If I change the order, amino acid 3, amino acid 1, amino acid 2, I'm going to get a totally different protein. So amino acid sequence matters, structure matters, and temperature and pH matter.